So these are two important signaling pathways. One is cyclic AMP dependent pathway and the other is IP3 oblique DAG pathway. So CAMP basically, as I told you, that it is a cyclic AMP. So it has two types. This pathway, CAMP dependent pathway, it has the stimulatory GS and GI, two different G proteins, and if GS is activated, it means it is going to start a process, and if the GI is activated, it means that it has to inhibit an ongoing process, right? And another difference between the two is that the target here is adenylyl cyclase, but here at GQ the target is phospholipase C. Another difference is that most of the time the GQ is involved with the lipids, right? But here you can see that there are no lipids involved. So these are actually the second messengers, CAMP and IP3DAG. These are actually the second messengers. As I told you that the primary messengers and the ligands themselves are the primary messengers, while these are CAMP and IP3DAG, these are the secondary messengers. So these secondary messengers, they are going to activate different pathways in different conditions, right? If here you can see if the adenylyl cyclase is activated, if it is a GI, so has to inhibit a process. If it is a GS, it has to stimulate a process, and the final product will be PKA, protein kinase A. So this protein kinase A will be doing some response in connection with these receptors. Okay, but here you can see that we have two different types of secondary messengers. One is IP3 and the other is DAG, and this IP3 and DAG, actually they are together as PIP2, right? And PLC, phospholipase C, is their target, and this target is then converted the PIP2 into IP3 and DAG, and this DAG and IP3 is involved with the activation of the calcium ions. The calcium ion is so important inside the cell, and the PKC, the the protein kinase C, it is again a secondary messenger, and it will create a response, in effect taking instructions from the activated receptors. Okay, so these are G protein coupled receptors. One of the pathway is CAMP dependent pathways, and the other one is IP3DAG pathways. GS, GI, GQ, these are different proteins. These are different G proteins, and they are responsible for the activation of different signaling pathways. Okay, so next slide is about that how actually it is activated, the G protein coupled receptor. So here you see this is the pocket for the ligands. The ligand will come here, it will bind, and then when in the absence of ligand, this G protein is attached with a GDP, right? And this GDP, it is attached when with the G protein, it means the G protein is inactivated. It is not going to perform any kind of function. Okay, so when it is activated, when this will be activated, when the GDP is replaced by GTP, and when it becomes active, it is going to instruct this phospholipase C, and this phospholipase C will be activated, and this activated enzyme, it is going to cause change in the PIP2. PIP2, as I told you, it is a combination of two different things, as we see in the next slide here. The PIP2 will be converted into IP3 and DAG. Right here, the alpha subunit of the G protein, it is activated, and after that the phospholipase C has become active, and then it is activated phospholipase C converted the PIP2 in IP3 and DAG. This IP3 it will go into the cytosol and it will trigger the calcium, and calcium is involved in hundreds of different processes. So cell will decide that how that calcium is gonna utilize in the cell, and then the DAG, it remains in the membrane, and where it activates protein kinase C, and protein kinase C further activates different signal transductions as required by the cell at that particular time. Okay, this is again the depiction of the formation of cyclic AMP. As I told you, cyclic AMP is the youngest sister of ATP. Here you see adenosine triphosphate, it has three phosphates. Then the adenylyl cyclase, this is the enzyme, it will remove two extra phosphate groups and it will convert it in a cyclic form. It will attach the ribose sugar. Here you see the ribose sugar, and this phosphate group will become cyclic. By cyclic, they will become more active, and they will get less space. And this is also secondary messengers, and because of its small size, it can activate lots of different signaling pathways within the cell. So again, this is a depiction of adenylyl cyclase, that when the GS, as I told you, that G protein that is responsible for the stimulation of a reaction. The reaction is not going on inside the cell, but once this GS protein is activated, this activation will lead to the initiation of a certain chemical process within the cell. Very simple. Okay, so this is, in this example, as you can see, that we have adenylyl cyclase. This is a ligand, it attached to the receptor, and after that adenylyl cyclase becomes active. When it becomes active, the ATP is converted into cyclic AMP. This cyclic AMP then will activate the protein kinase A. This protein kinase A is then going into the nucleus, the catalytic subunit, and the genes responsible for the activation of different processes will be activated here. So what the GI will do, I stands for inhibitory. So whenever there is a ligand bind to this type of protein, so what will happen? The process that already is going on inside the cell, it will be stopped. Right? Very simple. GS, it is going to stimulate certain reaction. GI, it is going to inhibit certain reaction. So GQ has different targets, right? Mostly it is involved with the lipid metabolism within the cell and the calcium metabolism within the cell. So here see the phospholipase C is activated upon the attachment of the ligand to the receptor, and then the PIP2 is converted into two different secondary messengers. One is DAG, the other is IP3, as I told you previously, and then the activated PKC, it will have different target proteins, and the activated calcium, it will have so many functions. Calcium, sodium, potassium pump, then we have calcium requirement for the fertilization, then we have calcium requirement for the bones formation, and for so many other purposes within the cells. Okay, so these are some of the examples of the signal transduction: GI, GS, GQ proteins, and they will use the 7 transmembrane receptors or the G protein coupled receptors, and they will activate different target functions in the cell. This is again a depiction of PIP2 and the conversion of PIP2 into diacylglycerol and inositol triphosphate, that is known as IP3. So the cyclic AMP and pKa, what is the relationship between these two secondary messengers? Here you can see that